Thanks. So today's topic is basically a continuation of what Skanda presented in the first webinar. That was on COVID-19 reinfection. So here we talk about COVID-19 vaccines. Now that vaccination is on in our country, the people have many questions. So idea of this presentation is to answer a few of the questions and also raise few questions. So let's say where we go. So basically when COVID started, I think within a month or two, it ceased to be a disease. It became a war. So we had Corona warriors, we had COVID warriors. So we stopped comparing COVID with any other disease. Such was the scale of the disease. And we have been fighting this war ever since. Now, who is our adversary? So our adversary is pretty small. It's a virus that we have named SARS-CoV-2. And why it's named SARS-CoV-2? It's because it is a successor to okay, just hold on. Right. So why it is called SARS-CoV-2? It is a successor to already known virus called the SARS-CoV-1. And obviously it is believed to have some zoonotic origins. Lots of debate about the origins. We are not talking about that here. But yes, on 20th January 2020, we came to know definitely that SARS spreads from human to human, and we know what has followed ever since. So this is a scene from one of Shakespeare's play on Julius Caesar, Et tu, Brute. And why did I put it? Since COVID is a war, we have to have few people who have made us fight this war. The humans were not supposed to get this virus if not for 3 human proteins. So those 3 proteins are, if you can see my pointer, so furin, ACE2, and TMPRSS2. Believe me, if we didn't have these 3 proteins, how you can bathe in a bathtub filled with SARS-CoV-2 and you would walk out fit and fine. So the ACE2 protein basically helps the binding of the spike protein, and TMPRSS2 and furin are the endogenous peptidases that help in the internalization of the virus. And what followed was deaths. And we are not talking of a few thousand deaths, we are talking of these many deaths. So over a period of, this is a data that I sourced last night from WHO website. So you see, these are not ordinary numbers. And for a virus that has spread for only a year, these are really extraordinary. But we have the vaccine, right? So what are the vaccines? And vaccines, many people think, are the elixir against this fight. So primarily, there are 3 vaccines that are being used globally. There are many local vaccines like Sputnik, Sinovac, Covaxin. Every country have their own vaccines, but this is basically a summary of vaccines being used worldwide. So one is the Pfizer, other is the Moderna, other is the AstraZeneca. In India, we are using this as Covishield, that is being manufactured in Serum Institute. So how do these vaccines differ and how are these made? So the vaccine is made from the genetic code of the virus. The genetic code is around 30 kb. Out of the 30 kb, around 3.8 kb codes for something called as the viral spike protein. Now we just learned that the viral spike protein is very important for the virus to enter into our body. So it's a gateway to the cells. So if we can somehow block the gateway, we can prevent the virus from entering into our body. So how is it made? Moderna and Pfizer have taken the route of the RNA molecule. So they have embedded the code for RNA in a molecule. It's basically an oily membrane-filled nanoparticle that gets injected into our body. Whereas the Oxford vaccine, they use a chimpanzee adenovirus, hence the name Shadox1. So there they use, the code is translated into DNA, and the DNA is then put into the adenovirus. This deactivated adenovirus gene acts as the carrier gene, and that becomes the vaccine for us. And how do the vaccines work? Yes, so once inside our body after a shot of the vaccine, these RNA molecules enter the human cell and they produce spike proteins in case of Moderna and Pfizer. In case of the Oxford vaccine, The DNA is first transformed into RNA inside the cell nucleus, and they make viral proteins. And these are spike proteins primarily. These viral proteins are again then taken up by the dendritic cells, which are the antigen presenting cells. They introduce it into our lymphatic system. Long story cut short, we have B lymphocytes, which produce the antibodies, and we have T lymphocytes, which produce, which destroy the infected cells in case we have an infection. So what does these B lymphocyte antibodies primarily do? Let's see, they are the neutralizer. So this is a spike protein. This is a small animation I tried to make up. So if you find under normal circumstances, they will bind with the ACE2 receptor and it will get into the cell and there will be infection. What if we block it with antibodies? Once we block it with antibodies, there is no binding, there is no entry, and we are protected. Game over. So we have won the entire battle. That's how we would like to believe it, but it ain't over till it's over. So let's go and understand why it's not so over. So this is the genomic organization of the SARS-CoV-2 genome. So this genome has primarily few parts, such as the non-structural protein parts, which produce very important molecules that is important for the survival of the virus. So we have RDRP, or the RNA-dependent RNA polymerase, the helicase, the 3CL protease, and papain-like proteases. So these are very essential for the virus to survive inside the host cell. We also have proteins like the spike protein, the envelope protein, the membrane protein, and the nucleocapsid proteins. These are structural proteins. They are very essential for the survival as well, but not maybe as essential as the non-structural proteins. Apart from that, you have accessory proteins that are encoded. And this has what has happened over the past 1 year. A virus that started off as a clade simply 19A, so this was the original mother strain of Wuhan, 19A, is slowly started diversifying. So as it started infecting more and more people, and as we started using more and more different types of therapies, the virus started mutating. And today we have more than 12 known clades of the virus. So no longer, we are not fighting a single virus now. And if you see how these clades are distributed around the world, you would see that their distribution also is not uniform. So few specific clade is present in more numbers. So if you look at China, you would find that the Chinese have more of the 19AB variants. But if you look at US, you see they have the 20G variant, the 19A variant, and every country have their own variants. So this data is taken from a website that I will be talking in a few moments from now. It's called NextTrain. It basically tracks, as more and more sequencing data are available, it basically tracks how the genomes are evolving over a period of time. Let's see one more thing, what has happened. Globally, if you find the distribution pattern of the clades of viruses. So here we were in 2020 Jan, mostly it was 19A, 19B. Now, as time has progressed, you would see the clades have kept on changing, and there is always a new emerging clade. One more thing that I would like you to understand is how the genes are changing. So this is the entire genome, and we will look at the entropy levels. If you look at the non-structural proteins part, the entropy is pretty low, so it's pretty stable genome. But when you start looking at the structural protein, the spike protein, you will see it's highly unstable. It's mutating pretty fast at the spike protein level. Our vaccine target is very unstable. And so is the other all structural protein parts. Because the virus can do without. I mean, even if the spike protein is not functioning properly, once the binding to the cell is done, the virus does not need the spike protein. So it can move on to infecting its host. And this is again a genome evolution rate. So you can see how fast the genomes are spreading. So we are not fighting against one virus. We are potentially fighting about 4,000 or 5,000 viruses as of now. The Sad part of the story is many of the countries are not sequencing the genomes sufficiently. So we might not know until a genome is widely spread, or maybe unless the phenotypic expression of the genome is somehow more, that why COVID is spreading more in one place and less in one place. And this is not all. There are two phenomena that happen with all viruses and even bacteria. That is antigenic drift and antigenic shift. So what are these? So antigenic drift is like if you watch superhero movies, it's like Iron Man version 1, Iron Man version 2, Iron Man version 3. Now, if you are but antigenic shift is something like two or three viruses come together and they put in all the parts together and come up with a addition of the virus that is entirely new, and there is a major antigenic change. So it's like Iron Man, Superman, and all those mans come together and they make a Wonder Man, and you really don't know what to do with it. And they have the potential to giving rise to pandemics. So let's see where we are. Today is February 5th. So don't get to the date. This is a I mean, this all these things happens. So this is actually February 6th edition of the Lancet. So the paper says resurgence of COVID-19 in Manaus, Brazil, despite high seroprevalence. So what does the paper say? The paper primarily tells us that in Manaus, Brazil, there was a huge number of COVID-related hospitalizations in early April, May, June of 2020. And then they did a serosurveillance study and they found, yeah, almost 50 to 60% of the population in Manaus, Brazil was infected with COVID, either symptomatic or asymptomatic, which primarily means they are now immune to COVID. But not so. So when the physical distancing was eased and local elections happened at around November 2020, there was a spike in number of hospitalizations. So hard was the spike that they had to go for a second lockdown. And you know what? When they started sequencing, they found that there is a new COVID lineage that's called the SARS-CoV-2 P1 lineage. So what we understand is, as we suffer from COVID or as we do vaccinations, we are immune to one variant of the COVID. We might not be immune to the other variant. Manaus is an example. What are the strains that are circulating now? So this is the most famous UK strain. So UK strain triggered a virtual lockdown in UK in the Christmas, as well as the flight services from the UK to and from UK. I think it's still suspended. So what happens here is there are 17 mutations. So N501Y, P618H, HV6070 deletion, 4 more on the spike protein. Also ORF Q27 stop mutation, that is the outside the spike protein. So what happens with these mutations are that here it increases the rate of transmissibility. More and more, it's more transmissible than the original SARS-CoV-2. But again, these are very recent studies. So how much, 40% is a estimate, it's not the true data. It might change to 70, it might change back to 20. As of now, there is very little concern about the current vaccine efficacy. Next, we have the strain, or that we always called the South African strain. They have 21 mutations, and many of them are on the spike protein. And the sadder part is, in vitro studies have suggested there is a potential for immune escape, which primarily means that you might get vaccinated with Covishield, but if you get infected with this strain of the virus, you will get COVID. Also, here is the P1 strain that was found in Brazil. 17 amino acid has changed, many of them on the spike protein. And there are anecdotes of reinfections, but potentials for immune evasions, it still remains unresolved. So these are very new reports. So scientists also take some time to understand how the immune evasion works. So we will propose a omics technique that can do it really fast. So this is another strain from the US, another Midwest variant again from the US, again one more strain from US. Now don't think that US is the center of COVID. It's just that US is doing far more many sequencing to know that the strains exist. Maybe we have a far more dangerous strain existing somewhere in some in Africa or some other country where they are not doing the sequencing and they might not even know. So this moral of the story is, from the what we understand from our history, is that humans are prone to get repeatedly reinfected with the common cold coronaviruses. So this is not the first coronavirus. Obviously, it's called the novel coronavirus because it's of a new kind. But there are other coronaviruses like 229E, OC43, HK1, NL63, and we tend to get reinfected by the same guy over and over again. So there were serological studies done, and people have found that 229E kind of attacks every 2 to 3 years. So we are not immune, or even if we are immune to the coronavirus, we lose our immunity to coronavirus. So what people did, they did a study. This is a time machine kind of a study. So the people went back in time. They used stored human sera from 1980s and 1990s, and they did a neutralization assay. What they found was when they used older human sera from the 1980s and 1990s, and they tried to neutralize a future virus, there is a virus that came in 2000 perhaps, these serums could not effectively neutralize it. So 18 to 17 years later, the neutralization part is lost. And the reason they found was due to the antigenic evolution of the viral spike. And what they advise at the end of the paper is, it is advisable to periodically update SARS-CoV-2 vaccines. So how do we do that? To win a war, we have spies. So we have to have spies on the ground to win this war against COVID as well. So we have two very good spies. One is Genomics, another is Proteomics. Let's see how they do the. So Genomics, next-gen sequencing, it has been most helpful. So the SARS-CoV-2 RT-PCR tests, we owe a next-gen sequencing. Very rapidly, the virus was isolated, and people did their sequencing. People designed RT-PCR primers. People designed all kinds of studies. They did a phylogenetic analysis, all thanks to next-gen sequencing. Now, if this would have been 1953 or 1977, in 1953 wouldn't have any idea what this bacteria or virus is. Far more people would have died. Even if it would have been in the 1980s, scientists would take around 6-7 months to sequence the virus. But thanks to modern technology, you can actually sequence the entire viral genome in a day. So this is our weapon to fight against this pandemic. And if you really want to see how this weapon is still being used, you need to go to these tools. So there are many influenza surveillance and response systems that are being set up in the world. One of them is GISAID, one is GSIRS, GISRS, and another is NextTrain. So NextTrain is a very wonderful website. Maybe someday we can have a talk specifically on NextTrain and how it can help you to track different diseases. So they have viruses, even not only viruses, they also track tuberculosis, measles, dengue, mumps, Zika, West Nile virus, seasonal influenza, and SARS-CoV-2. They have started tracking as well. The name is NextTrain because they were born to predict. They were given, the first job they got was that you have to predict what is the next seasonal influenza strain against which we should vaccinate our population. So although not very popular in India, in the US it's very popular that people would get seasonal flu shots. So influenza vaccination will be done. That is something regularly done. NextTrain guys predicted using genomic data that which strain of influenza would be the most prevalent strain in the upcoming season. So this data was then transferred to the people who designed vaccines, and the people who designed vaccines started designing based on the inputs from next strain. It's very interesting how they do it. You can spend some time on this website. Next is proteomics. So in proteomics, one of the beautiful experiments that we can use or tool we can use is protein microarray. What does protein microarray do and how it can help us? So this is basically what we can do with the protein microarray. A person is getting infected by SARS-CoV-2. So the virus will enter into the cell and it will produce it will start replicating inside the cell, and finally it will produce, our human body will produce antibodies. Now, if the human antibodies, if we can use a proteome microarray, we can actually understand or do the epitope mapping that where exactly, which part of the viral proteome we are using is generating the immune response. So let me give you give it up in detail. So the proteome microarray, this is basically a commercial design. In this commercial design, there are 15 amino acids. So the entire SARS-CoV-2 proteome will be printed on a glass chip. And there are 15 amino acids each. And they are printed in an overlapping manner. So there will be a 13 amino acid overlap. Now imagine if the human body produces a IgG against a specific SARS-CoV-2 peptide, antibody will automatically recognize its epitope. So this is what we call epitope mapping. So the epitope will bind with onto these arrays, and we can find that, okay, this human being, this particular patient, has actually generated a IgG that is using the antigenic region of this specific antigenic region of the SARS-CoV-2 proteome. So what we will see primarily, what we can do is, people, once we identify the people that these are the specific group of people having a COVID infection from a different strain of virus, we can immediately take their serum and then test for the epitopes that it is producing antibodies against. If they are producing epitopes against known, if they are producing antibodies against the known epitopes, the epitopes that also the vaccine targets, then it's fine. We can tell that, okay, the vaccine should be working for the general larger population. But no, if they are producing a specific epitope that is different, then we can also give them the makers of the vaccine, a new target, that you should now try to target your vaccine design in such a way that the epitope for specific clade should be this. So each group will have a group specific epitope. But not all is lost. As of today, there is no reason to worry, because India's Covaxin, Moderna's, and even Pfizer's vaccine, they have people, very preliminary research has been done, but they are seeming to work, especially against the UK and the South African variants. But as I said, that we should be always very aware of our surroundings. We should keep on this genomic and proteomic surveillance. So once we put on our surveillance cameras, we have to keep on tracking where mutations are coming, how the mutations are behaving, if the disease cause, is it the severity is increasing, or is the antibody response, if already vaccinated people are getting COVID again, that's a reason for worry. So we have to keep up our guards and we have to keep fighting this war. So thanks are due to Professor Sanjeeva from our lab, and also our lab members. It's a very competitive lab. People are always reading new stuff, so they are always keeping on your toes. So we have to understand more and more of of what's happening in COVID biology. Dr. Radhika Vaishnav, she is very encouraging. Helped us set up this entire omics boot camp. I mean, it is her brainchild, sort of. She pushed us. Otherwise, we are very. I'm a very difficult man to push and get things done. Amartya Nandi, Gitanjali Ghosh. Gitanjali, thanks for moderating the session. Amartya, thanks for making those flyers. and all members of the Omics Bootcamp. So we are in this together and we will get through this together. Thank you for your attention.
All right. So you have a question from Abhishek, I think. Let's
Just
see.
let me.
Okay. Yes, if PCR tests are easily contaminated, how sure are we about the accuracy of the case numbers?
Can you, if PCR tests are contaminated, let me see the question. PCR tests are not contaminated. Like, I don't know what Abhishek means by getting a PCR test contaminated. Usually, PCRs are done with lots of care. Now, what happens is in case of sometimes due to the mutations, the primers that detect these genes, and this is a real world problem. So during the COVID test, usually the one that we do for PCRs, what we do is we take target very specific gene segments. We don't check for the entire viral genome. We check for the spike protein, we check for the nucleocapsid protein, and stuffs like that. Now, and if you know if how PCR work, we need the primers to efficiently bind with the region that it is trying to amplify. Now, just in case there is a mutation in the binding region, you would find that people who are positive for a specific virus for COVID, they would also turn out to be negative. Reason is the binding of the primers would be improper, and it will give a very high CT value, more than 40. This actually happened for the UK strain, and there's a very nice story how UK strain got people discovered. So people were using Thermo Fisher TACPath. Now TACPath has three targets for the gene. One is spike, one is E or the envelope, and then another is nucleoprotein. Now, suddenly they found a group of people who were falling sick, but their spike protein reports were coming as negative. They were positive for N and E, but their spike was turning out negative. And this is the reason was that the spike protein probe binding region had mutated. And so although they were having COVID, they did not give any response for the spike protein. So so that's why whenever we design a PCR test, we always keep in mind that we have to take at least two genes that is specific to the virus. Yes, Abhishek, you want to say? I mean, that's what I understood from your contamination. You can go ahead and ask your question.
Sir, I have found that PCR is a gold standard platform. Hello, sir.
Yeah, I can hear
Hello.
you. Please go ahead.
Yeah. Sir, you have also told that PCR is a gold standard platform for testing, but even a teeny amount of virus in a patient sample can be found and amplified for detection and testing, and that does not mean the test is foolproof, right? So the results can be easily contaminated, which is precisely the CDC's initial rollout of coronavirus test that was found in February. Also, they have found that many of the negative control tests are faulty because. Some of the reports are also said that these tests are going to be false, and um, it may have some various reasons that why these tests are faulty or why this misconduct happening. But can you please tell us that how PCR test can be rightly proved? Means how can the test can be authenticated?
You cannot authenticate one PCR with another PCR. I mean, that's
Okay.
but we can obviously marry technologies. Like we can use another technology like seroprevalence studies. We can use
Yeah.
lateral flow assays. Now people have coming up with these rapid antibody tests that people are using for nucleoprotein and other technologies. Obviously, we can obviously go for next gen sequencing if it is feasible to actually check. And then obviously you have to look at the clinical parameters. Now, there are a few people who are immunosuppressed, and they tend to they take a very long time for the you know clearance of the virus. So they will give you COVID positive results for as long as a month after infection, but the virus would have been cleared. Sometimes the viral nucleic acid particles are persistent in areas like the gut. So yes, you can get negative results, but obviously you have to marry it with other results like how is the CT coming, how's the chest CT coming out, whether the
Okay.
first patient has turned seropositive. and you cannot get like a 100% foolproof. Like any technology will have a error. So you can take a 99 or 95% as a very good way of turning specific, and then you can take 4 5 other tests. There's somebody who is having ground glass opacities in the lungs, is having difficulty in breathing, is also turning SARS-CoV-2 positive. They have to be a COVID patient, right?
Yes. So, sir, in our India also these cases are found because the technologies to technology it may vary. That's why this is the main reason that you want to tell us, right?
Oh, yeah,
Means
India is
technology
also
to
doing a lot
technology.
of
Yeah.
sequencing, but we are sequencing less. Given the population, we should sequence more. But yes, we are doing good compared to other nations, but maybe we can never match up the prowess of America. They have a much better infrastructure than us. Somehow they also seem to have many more cases than us. So India, the caseload has come down significantly. But yes, we have to keep our eyes and ears open. If we see that in a specific area there is suddenly a spike in the number of positive cases, that is a reason for concern. We should immediately go for a next-gen sequencing-based approach, sequence the virus, check its phylogeny. Then we can also go for these microarray chips, check for the patient's serum, and check if their antibodies, what kind of antibodies they are generating. Are they generating the same kind of antibodies like the person who was maybe from some other person who was 6 months ago, or their epitope profile has changed? If their epitope profile has changed, it will immediately tell us the virus has undergone a big antigen shift. So yes, we have to keep on we have to keep our guards up. There is no way or other way of doing that. And yes, perhaps 2 or 3 years down the line we will know more about the virus. So we have just completed a year uh, that with this virus, and
Yes, sir.
we barely know. I mean, this is one of those subjects that have been studied at a very rapid pace. The number of
Yes, sir. Yes.
publications you find on COVID easily beats if you check for other diseases like cancer and stuff. So what cancer
Yes.
research was done in 5 10 years, COVID has done in a year.
In
So,
just
but still, we need to know more because few things take time. Whether the immune memory is working, there is no way other than to wait and watch. So more and
yeah,
more
yeah.
people are getting vaccinated. The idea is less and less people should get COVID, because if the virus enters a cell, it will definitely mutate,
Yes, sir. Sure, sir.
and
Thank
we
you
do
so
not
much,
know
sir.
how that mutation will work.
Thank you so much, sir. It was a very nice presentation presented by you, and we are extremely delighted to meet you in this presentation, sir.
Thanks.
Thank you, sir.
Okay, all right. Is sir there? Sanjeev sir?
Yeah,
Or
I'm
I
here, but
should?
I think Argyo has one more question. Maybe you can take one.
No, just to add on to Dr. Acharjee's lecture, like Abhishek Chattar was, like when we are developing a test, either it is an PCR based or an antibody based, we generally go for the sensitivity and specificity of that assay. So how much my test is powerful, you go by that. There is a chance that 1% through RT-PCR also the test gets corrupted, means due to the false positive ratio, but 99% it is specific. No other test in the world is as specific as RT-PCR. So that is why it is called as gold standard. Either you take the ELISA one or you take any of the spectroscopic methods. So this is an addition. So there is a chance that 1% of the population will be false positive for the SARS-CoV-2. That's all.
Okay. Anyone else?
All right. So given the time, I think we can keep it 1 hour, and we can definitely have more questions in our Telegram group or any other forum to ask the presenters. So Arup, thanks for making very nice presentation. I'm sure everybody have found very informative, and both the presenters, in fact, Joshua and Arup, did good job of conveying the latest updates in the field. Let's continue this webinar series. I think Dr. Radhika, and I'm also finding interesting that there are more and more volunteers coming up who are helping to improve the quality. So Dr. Seema help mentoring Joshua, which was very good. And I think we need to see more faculty and more of the senior people to mentor these students and give them more opportunity to improve their skills in this particular space. I think that's all for, today. Anything else you want to add, Gitanjali?
No, it's okay. Just a thanking note from us. Thank you to our journal team, whole journal team. I mean, hats off. All are working so hard and so good. Their contribution are just inexplicable. That's and thank you all, and thank you, sir and ma'am, for giving us such a platform, such an interactive platform. Thank you.
Wonderful. So we'll see you all in next 2 weeks time again. Let's look for more speakers to come forward.
Yes. Yes.
Okay. All right. All the best, everyone. Okay.
Thank
Bye.
you.
Thank you. Thank you, ma'am.